Good morning students. നമുക്കിന്ന് പോളിസിഫോണിയയുടെ റീപ്രൊഡക്ഷൻ നോക്കാം. അപ്പോൾ പോളിസിഫോണിയയുടെ ജനറലി സ്പെഷ്യലി നമ്മൾ ഇന്ന് നോക്കാൻ പോകുന്നത് സെക്ഷുവൽ റീപ്രൊഡക്ഷനാണ്. അപ്പാർട്ട് ഫ്രം സെക്ഷുവൽ റീപ്രൊഡക്ഷൻ they also have vegetative and asexual modes of reproduction. Vegetative mode of reproduction, it happens through fragmentation, and asexual mode, it happens through by means of spores, and these spores may be of several types. This polysiphonia, they are having three different morphological forms, or three different forms are being there as it completes its life cycle. The three forms are gametophyte, carposporophyte, and Tetrasporophyte. Okay, so the gametophyte is the haploid and the independent plant body. Carposporophyte is the diploid form, but it is not independent, it is attached to the female plant, it is dependent on the female plant. And the Tetrasporophyte, it is a free living diploid plant. It forms by the germination of carpospore. Carposporophyte produces carpospore, and that carpospore germinate to form Tetrasporophyte. Okay, and in Tetrasporophyte they form haploid tetraspores. These tetraspores, they germinate and produce the male and female gametophytic plant. So this is an overall briefing of its life cycle. So let's move on to the sexual reproduction. In sexual reproduction, there are male and female gametes are there, and these are formed in respective sex organs. Here the reproduction is oogamous type. Polysiphonia is a heterothallic species. So male and female sex organs are formed on different plants. Male and female plants are there. Male and female thallus are there. And coming on to the male sex organ, male sex organ is known as spermatangia, or it is also known as antheridia. Okay, male sex organ is spermatangia or antheridia. It is born in clusters on the male plant. You can see here this is an antheridial mass. It is born in clusters, and this is a male plant. It is mostly produced on the upper end of the dichotomously branching trichoblast, and the two basal cells of the trichoblast, they function as the stalk, and the other cells, what they form is they cut off certain cells. They cut off pericentral cells. See, this is the pericentral cell. This is the central siphon and or the axis of this cone. It is a cone shaped structure, and this is the central axis, or the cells which form the central axis, the siphon of this cone. From this cell, certain cells are being cut off, that is known as pericentral cells. So these are the pericentral cells. Okay, so pericentral cells are cut off, and the this portion, this fertile portion, that is this portion, it becomes polysiphonous. Now this pericentral cells, what happens is, these pericentral cells, they can divide once or twice. It may divide once or twice and produce a large number of spermatangial mother cells. These are the spermatangial mother cells. And each of these spermatogenial mother cells, it give rise to 4 antheridia. Okay, so once again I repeat, male sex organ is known as spermatangia or antheridia. It is born in cluster. As you can see here, it is seen in cluster and it is born on the male plant. Male plant has a branch known as trichoblast, and this male sex organ is born on the upper part of the dichotomously branching trichoblast. Okay, the central axis of this cone shaped structure, this antheridia or this spermatangia is a cone shaped structure. The a siphon like region is there, a siphon like central axis is there, which cuts off pericentral cells. These pericentral cells, they cut off certain cells which form the spermatangial mother cells. These spermatangial mother cells, they give rise to 2 or 4 spermatangia or antheridia. Okay, that is all about the male sex organ development. And after the formation of this antheridia, what happens? The content of this antheridia produce a single non-motile gamete. From each of these antheridia, they form single non-motile gametes, and it is liberated through a narrow slit, and it is carried through the water current towards the female sex organ. And now let us see about the female sex organ. Female sex organ is called as carpogonium. Female sex organ is called as carpogonium. It is born on a filament. A 2 to 5 celled filament is there, which is known as carpogonial filament. See, this is the carpogonial filament. So it is born on a filament known as carpogonial filament, which is 2 to 5 celled. Carpogonium actually is a flask shaped structure. Okay, it has a swollen base and an elongated neck-like region. This neck-like elongated region is known as trichogyne, and the egg or the female gamete is present in the basal swollen region. Okay, so during the development, one of the pericentral cells which is cut off from the trichoblast, this pericentral cell of the middle tier, it functions as a supporting cell, and it divides again to form a 4-celled curved carpogonial filament. This is the carpogonial filament. Okay, it is formed from the basal cells of the supporting cell. The supporting cell, it divides several times, 2 or 3 times, to form the carpogonial filament. The terminal cell of the filament, it becomes the carpogonial mother cell. This terminal cell, it becomes the carpogonial mother cell. See, this terminal cell, it becomes the carpogonial mother cell, and it differentiates as carpogonium. 2 basal cells of the filament, it becomes 2 sterile cells, and they are known as basal sterile initial and lateral sterile initial. So this is the fully formed trichogyne. And after this development of female sex organ, that is the carpogonium, fertilization takes place. The male gamete, that is the spermatia, which is liberated from the slit on the male sex organ. It is liberated to the water, and through water current it moves to the vicinity of female sex organ. The wall at the point of attachment between the spermatium and this carpogonium, it dissolves, and through this opening, or through the point where the wall dissolves, the male gamete enters into the trichogyne, and here it reaches the, it approaches the egg and fertilization happens, and it diploid, that is the zygote is formed, and this is the diploid phase. Okay, so this is the diploid phase. That is how fertilization happens. Now after fertilization, certain post fertilization changes happen. That is the formation of Tetrasporophyte. The fertilized carpogonium, it faces several changes after the fertilization process. The first change is the formation of an axillary cell. The supporting cell, which was formed earlier, a cell is formed beside this supporting cell, just close to the carpogonium. The axillary cell, which is haploid, it establishes a tubular connection with the base of the carpogonium. Okay, what happens? The axillary cell, it establishes a tubular connection with the base of the carpogonium. Okay, and the diploid nucleus of the carpogonium divides mitotically into two. Of the two, one passes into the auxiliary cell through the tubular connection. The haploid nucleus of the auxiliary cell, it now degenerates, and only the diploid nucleus remains. Okay, the pericentral cells, which are close to the supporting cell, what they do is they produce various filaments which envelop the entire Tetrasporophyte. So like this, these vegetative cells, they envelop the Tetrasporophyte. The carpogonium and trichogyne gradually they start degenerating. Now certain initials known as gonimoblast initials are produced. Okay, and each initial divides and redivides to form a short gonimoblast filament. The terminal cell of the gonimoblast filament, it swells and it functions as carposporangium, and the contents of the cell differentiate into single diploid carpospore. Okay, now the whole structure consisting of the gonimoblast filaments, the carposporangia, the carpospores and placental cells gets completely enveloped by the vegetative filaments and form an urn-shaped body, which is known as cystocarp or carposporophyte. Okay, cystocarp or carposporophyte. Its sheath is known as, its covering is known as pericarp. It has an opening, and that opening is known as ostiole. Okay, now next is the formation of Tetrasporophyte, which is a free living plant body, and it resembles the gametophyte in its appearance. So, but as it is this asexual plant, it does not bear any spermatangia or any other female sex organs like carpogonium. But instead of it, they bear a structure known as tetrasporangia. The diploid nucleus of each tetrasporangia divide meiotically and produce 4 haploid tetraspores. And the tetrasporangia are formed from the small sized pericentral cells. And now what happens is, these pericentral cells, they divide vertically, and an outer cell and an inner cell is formed. The outer cell, it divides and forms 2 or more cover cells, whereas the inner cell, it functions in a way to form the sporangial mother cells, and it divides to form a stalk cell and tetrasporangial cells. Tetrasporangial cell, it acts as the tetrasporangium, and the nucleus divides to form, meiotically divides and forms 4 tetraspores. These tetraspores, they germinate and produce the haploid gametophytic plants. It forms the male and female plants. Independent plants are formed. So this is the life cycle. This is the male plant with spermatangia, the cone-shaped spermatangia. This is the cross section of this cone-shaped spermatangia. And this is a female plant with carpogonium. And then what happens after fertilization? Cystocarp is formed. In the carpospore is formed. From carpospores, it germinates to form the tetrasporophyte, and tetrasporophyte produces tetraspores. These tetraspores go and germinate to form the male and female gametophytic plants. So this is all about the life cycle of polysiphonia.